Thank you, everyone, for coming here. I really am excited to be here. Thank you for the University of Chicago for hosting me. I love Chicago. I love the city. So we were talking, any excuse to come here, I'll take it. So like Valerie was saying, this work is part of that pilot grant that it wouldn't have happened without the grant, because as I'll go through with how expensive data can be and how expensive data workshops can be to understand how to use the data, it was really instrumental in getting this project off the ground. And so today I'm going to be talking about epigenetics, specifically epigenetics and how it relates to adolescent depression. And I think epigenetics is one of those words or one of those things to study that is very popular. It's kind of like the hot topic right now to study, and it's very exciting. Everybody wants to do it. But I think a lot of people don't know exactly what it is, what the molecular mechanisms are, and actually how to study it, what statistical methods to use, and things like that. So today, this talk is largely going to be about the background concepts of epigenetics and how we can apply it to understanding development, and also a lot of the methods and data gathering that goes into epigenetic studies. So hopefully when you leave here, you'll have a good understanding of what epigenetics is and also how you can study it if you want to do a study with epigenetics. So I just want to talk a little bit about my background, because I'm a child clinical psychologist, so I do a lot of treatment. And so a lot of people ask me, how is someone who is a therapist, how is their research about epigenetics, which is very basic science, right? And epigenetics is kind of a combination of all the things that interest me about development. So I'm interested in child development, specifically development over time. So longitudinal models. I'm interested in sensitive periods of development, what periods of development are important for certain outcomes. I'm interested in internalizing disorders, so childhood anxiety and depression. I'm interested in how genes and environments interact with each other to confer risk for some of these outcomes. And even in my therapy, I'm interested in the biopsychosocial model. So I work in a lot of integrative settings in hospitals and primary care practices where I work with children who have chronic disorders, who have a large biological component to what they're struggling with. And so also in my clinical work, I like to think of development at the whole. And of course, I'm interested in epigenetics. So this is part of my dissertation, and it's a culmination of all the things that I am interested when it comes to child development. So before I go into the minute details of epigenetics, I kind of want to give you guys an overview of what it is and how we think about it. So very broadly, epigenetics is any process that changes gene expression but doesn't change the DNA sequence. So our DNA sequence, the 23,000 genes that we inherit from our parents, they're not changeable. That's the sequence that we get. And so in the past, people would think that genetics is this really stable thing that's deterministic, that if you have this gene, you're going to have this outcome. But with epigenetics, there are markers that are on top of the genome that turn those genes on and off. So yes, we can't change what genes we inherit, but we can change how the genes are working. And the best way I heard it being described once is if we think about it in terms of an orchestra. So an orchestra is the outcome, right? That's the phenotype. That's depression, that's anxiety, that's cancer, that's any kind of outcome that you're looking at. It's when all the pieces come together. It's all the different instruments, it's all the conducting, and everything, that's the outcome. The sheet music would be the structural genome. So that's the genes that we don't change. That sheet music, for whatever score the orchestra is playing, stays largely the same. And that represents our genes. But the epigenome is the layer that goes above the sheet music, the markings. So when certain conductors want certain music to be played differently, they mark up the sheet music. And that's kind of what represents the epigenome. And the red ink are the permanent markers. That's when the conductor decides, I want this to play this way all the time. And those are the permanent and stable changes of epigenetics, and I'll go into what that is. But the pencil markings, the ones that you can erase, that you can add on, that you can take away, that's the epigenetics markings that people are interested in, because they're changeable in response to the environment, and they're also reversible. And so that's why people are really excited about epigenetics, because these pencil markings that can lead to certain types of diseases, both physical and mental, can be reversed, can be erased, or added on. And so if we first talk about the permanent marking, so the red ink, the stuff that doesn't change, every cell in our body contains the same genetic code, has a nucleus with all our DNA in it. But how can we have different cells? How can we have different tissues, right? We're not all a massive glob of the same genome. Certain genes are being turned on and off in every cell. That's how we have a liver. That's how we have a heart. That's how we have lungs. Because epigenetics turns those genes off, right? So if you think about the permanent epigenetic markings as like cell differentiation, it basically kind of runs in the background, turning certain genes on and off so that we have different structures and different functions to those cells. And so this is essential for normal development. Otherwise, we would just be like one clump of the same types of cells. And so this doesn't change much from person to person. If we look at our genomes, when we talk about these markings, they're largely the same in humans. And these aren't really reversible. These are the stable changes that epigenetic usually does in prenatal development. But with cancer, this is when these permanent markings are being changed. So that's why cancer is such a difficult disease to treat, because it's actually changing these permanent markers. And so every cell has these tumor suppressing genes that are constantly being activated, right? So these are genes that are coding for proteins that suppress tumors. With epigenetics in cancer, something comes in and shuts those genes off, and then the tumors start to form. The malleable markings, when it comes to development, is something that I'm interested in. So the malleable markings, researchers have found, are very sensitive to the environment. And environment can be thought about in many different ways. Some people study the environment in terms of nutrition, how certain different foods, especially in the prenatal period, change epigenetic markings. Some people, like me, study stress and trauma and child abuse and how those experiences change the way that layer is functioning. And some people study cigarette smoking and alcohol and things like that, and how that changes the way that genes are being turned on and off for certain things. So the actual molecular process of how this is working, it's called DNA methylation. There are many other different epigenetic mechanisms, histone modification and microRNA, but we're not going to talk about those, largely because people don't really study them. Most epigenetic studies, or if you see in the news or on media that this study talks about epigenetics, is usually about DNA methylation, because DNA methylation is cheap. It's easy to get. You can get it through blood. You can get it through buccal cells. You can get it through saliva. It's very easy and non-invasive. And it's what we understand the most when it comes to the molecular structures of this. So we have a really good grasp of understanding how DNA methylation works. And basically, if you look at this is your genetic code. These are the 4 basic nucleotides. And what happens in DNA methylation is a methyl group, which is CH3, binds to a cytosine before a guanine. And it binds to where that nucleotide is located, and then it either activates a gene or it shuts down a gene. In the past, people would call DNA methylation just complete suppression. So before, when technology wasn't as good, when it came to DNA methylation, we thought of it as largely a suppressing mechanism, but now their studies show that it can activate genes as well. So 5% of the cytosine bases that we have in our DNA have these methyl tags on them. And the way that these things are located in our genome is they're called CPG sites. So it's basically cytosine and then the guanine. And they're called CPG sites, and they're largely found clustered together. And the reason why this is because it's evolutionary adaptive, because you have this part of the epigenome that's highly adapted to the environment, really susceptible to changes. You don't want it to be easily accessible. You don't want it to be everywhere in your genetic code. So they're basically found together in these large clusters, and they tend to be unmethylated. So there are no methyl tags on them. And they are often found in the promoter sites of genes where transcription happens. And it's associated with a gene that it's always transcribed. So when I was talking about the permanent red markers, these are the CPG islands. And so when you're thinking about cancer, it's the CPG islands that are starting to get the methyl tags and starting to get shut down. So the housekeeping genes that maintain the processes in our body are being shut down, which is bad. But what researchers now think is important is going out from the CPG islands, the shores, the shelves, and then the open sea. I think they started with island and they went through the whole metaphor of shores and the sea. So the CPG shores are less dense and they vary across individuals a lot more. So these sites are pretty much the same between me and you. But the island shores, that's when we vary differently among individuals, and that's where researchers think that individual differences can be found. So we really want to study those CPG shores and shelves and going outwards.
Can you clarify?
Yeah, go for it.
So you mentioned that the CPG sites are, there's an evolutionary advantage for having them unmethylated, and not being subject to or other epigenetic processes. True?
Yeah, the ones that.
Yeah.
Okay.
So I guess I don't understand how this logic doesn't. So you mentioned cancer. So I mean, many times when there are epigenetic problems that are persist, it's a problem. It's not about the evolution didn't find the optimal solution, right? Or did it? I mean, I guess I need the clarification.
Yeah. No, I mean, the way we usually study when epigenetics goes wrong, right? We study it when it causes diseases, but epigenetics changes in and of themselves aren't necessarily bad. Because, like, for example, if you exercise, you are changing the way that your epigenome is working for the better. So it's not necessarily that any kind of epigenetic change is bad. It's that, so that's when we're talking about here, but the CPG sites, this is where change tends to be bad because these are supposed to be typically unmethylated. So yeah, I don't really know the evolutionary argument. I'm not an evolutionary person, but that's what people think, why they're not found everywhere. They're found in clusters together, and they're rare in the genome. And so, right, this is very basic science. And so how did I get into it? So the interesting thing about epigenetics, and with epigenetics, I think a lot of times what tends to happen is researchers get really excited and they tend to leap before we have a lot of evidence. So a lot of these processes are much more complicated than I'm making them sound, and a lot of the stuff is still theoretical. I like to say that epigenetic research is still kind of in the dark ages, and work in this is pretty much incremental, and as technology advances, I'm sure a lot of this is going to be turned on around on its head. But the reason I study it is that developmental researchers are thinking about epigenetics almost as a mediating factor. And so we know that environment affects disease, and specifically for someone like me who studies depression and anxiety and things like that, there's a lot of evidence for lots of years how life stress, trauma, abuse leads to things like depression and anxiety and things like that. So we know that's true. And epigenetics is maybe an explanation of how biologically it's true. So it can get under your skin and change the way that certain genes are working that can start these disease trajectories. And so the first real study that tested this model was in rats. It's an animal model. But it's a really good example of the possibilities of epigenetics. So in 2004, Weaver, they took rat moms and their pups. And so in rats, like in humans, there's a natural variation in nurturing. So some rat moms are super nurturing. They lick, they groom, they feed their pups, and some moms just naturally aren't. They don't lick a lot, they don't groom, they don't feed them as much, right? And what they found, like we find in humans, less nurturing moms, higher levels of anxiety and depression, that's what they found in these rats. Just naturally occurring low nurturing mothers tended to have pups that are more anxious. And when we talk about anxiety in animal models, we basically mean things like freezing behavior. So when rats get anxious, they freeze. And more anxious rats tend to freeze a lot. When they hear sounds, when they see lights, things like that. They're also less likely to explore. So if you put food in a dark maze, they're not going to go to it. But the less anxious pups will explore. So that's how we think about anxiety in terms of animal models. And so they wanted to see if DNA methylation has anything to do with it. They picked one particular gene called the glucocorticoid receptor, and basically that codes for the glucocorticoid genes. And the glucocorticoids are responsible for shutting down our body's stress response. So our stress response is very adaptive in that when we're faced with danger, we have a fight or flight signal. But that signal needs to be shut down once the danger is over. Otherwise, it's very taxing on the body, it's very taxing on the brain. So they were looking to see if these glucocorticoid receptor genes were being shut down. Meaning, were they being shut down? Did these pups have an inadequate shutdown of the stress response system that leads to anxiety? And that's actually what they found. They found this mediating model. So low nurturing mothers tended to have pups that had higher levels of methylation in this gene, which was related to the level of anxiety they were showing. And high nurturing mothers had lower levels of methylation, so that gene wasn't being shut down and were less anxious. So this is a really cool way to see how epigenetics can fit in between life stress and a certain type of outcome. Again, this was just one gene. All our genes are working together. It's a very simple study, and it's really difficult to do in humans, but it kind of serves as an example for what epigenetics can do. And the interesting thing about this study is that they found that the epigenetic changes were reversible. So when they added the methyl group tags to the pups, they changed the behavior. So the anxiety pups now were less anxious when they erased the methyl tag. So now the gene was being expressed. And then they took the non-anxious pups and they added the methyl tag, which suppressed that gene, and then they became anxious. So it's kind of this cool way of how they reversed, they changed behavior through epigenetics. And so my project that I used this grant for kind of echoes that original animal model, because these types of projects aren't really done in epigenetics. So what typically people do is they look at either this side, they find some genes, or they look at this side, but they really never connect the three things together. And so if I'm interested in development and I'm interested in how things develop over time with DNA methylation, I want to look at it in a mediational model. So basically, I'm doing two separate analyses, and I'm doing something called an EWAS. So an EWAS is an epigenome-wide scan of DNA. So if you're familiar with a GWAS, it's the same principle. So in a GWAS, they take a group of people who have schizophrenia and a group of people who don't have schizophrenia, and they scan their genome to see what genes are different, what genes have different SNPs. And then based on that, they think that those genes might be involved in schizophrenia. We do the same thing, but we just scan the entire epigenome. So all the CPG sites have already been found and annotated, just like the genome has been annotated from the Human Genome Project. So basically, we scan the entire epigenome. And I looked at it in two separate sensitive periods of development. So there's a lot of interesting research about how a lot of stuff prenatally can predict psychopathology later in life in children. So lots of prenatal maternal stress. There's a lot of research that it predicts depression in adolescence in those children above and beyond anything else that you would think is related to depression. So beyond mother's own depression, beyond SES, beyond life stress, something that happens prenatally is really important later down the road. So this model is going to look at maternal stress, scanning the epigenome-wide, so methylation at birth, so we have cord blood data, and then seeing depression in adolescents. And then the second study is looking at adolescent life stress, because adolescent represents the second sensitive period, especially when we think about depression. Because depression in children is really, really rare, actually. I think the prevalence rates, are like 5%. It's really rare in childhood. And then when you hit 12 and 13, it kind of skyrockets. And there's a lot of explanations for why that happens, and it's probably a complicated mix of a lot of factors, including puberty hormones, and peers and things like that. But I wanted to study that period because that's when we see depression tend to skyrocket. So I looked at adolescent life stress, scanned the epigenome when they were 15 or 17, and depression in adolescents. And the other strength, I think, of this project is that depression in adolescents is measured longitudinally. So it's not one time point. Basically, I have 4 time points starting from 12. So before that kind of skyrocketing happens, all the way up to 18. So we have 4 different time points of depression, and I made different depression trajectories, which I will go into. And because this is the whole of my dissertation, but this pilot grant in particular helped me get this part started and helped me get money to get this part started, so then I could do the whole model. So that's the part that I'm going to be talking about today.
So just to understand, metaphorically, you have data on an individual, and you know you're looking at how the markings on the score for the music changed in the same person across time. So you have a sequence of EWAS examinations of the same person. You can see where there are differences.
Yeah, so that, and I can see between individual differences.
Yeah, so I want to know about the temporal dimension.
Yeah,
You can see where the erasures and inclusions are on the score.
Yeah, and you could see, and I've had a lot of conversations with people about this. Does the DNA methylation change from birth to 15 or 17? And some people, make the argument that they should stay the same, that there shouldn't be that much differences within an individual. I actually think that I'm expecting them to be different, because I think maybe different genes are more important in different parts of development. So you can imagine in prenatal development, what genes are more salient might be the ones that are being methylated, whereas maybe in adolescence, there's different genes that need to be activated in that development. But I don't really know. So that's an interesting question to see about within an individual. Are these changes stable over time, or are they different?
Well, I mean, you brought up examples where you might think that's the case, and you think that there's issues of peer pressure and the other horrors of adolescence.
Exactly.
So you would look at the same individual, and what you would see is changes in the methylation after puberty.
Exactly.
I mean, is that something you have data that will, the principal, let you look at the dynamics?
Yeah. And I'll go into what the data is like. And so basically, a lot of the strengths of this study rely on the longitudinal design. So actually exactly what you were talking about, I can look at DNA methylation at multiple time points within the same person. And I can also look at depression over time, because there are EWAS studies of depression. They're usually in adults. They pick one time point and then they do an EWAS, which to me, like if I'm interested in looking at how depression develops, I think this is a better way of looking at it, especially because when you're looking at someone who's in their 40s, this is probably their 2nd or 3rd depressive period in their life. So I want to look at when it starts to see if there's any epigenetic markings going on. And so to study something like this, you need a really good data set. And I used ALSPAC, and I don't know if you guys are familiar with ALSPAC, but it's a really, really awesome birth cohort in the UK. Basically what they did is they picked a town called Bristol in England, and they tried to recruit every single pregnant woman in that town. And they managed to recruit around 72% of the population, which amounted to almost 15,000 pregnancies. So they had these 15,000 moms, and then they followed these children pretty much indefinitely. So they're called children of the 90s. Right now, I was born in the 90s, so they're about my age. They're in their late 20s, and data is still being followed on them, and now they're children, and they plan to keep going. And it's really cool because they have so much different assessments that whatever you want to do, you can pretty much use their data. So they look at stuff like BMI, nutrition, school, friendships, romantic relationships, stress, parents' employment, like everything you can think of, they look at. And they also have good measures of psychopathology. So someone like me would be interested in anxiety and depression and things like that. And they have pretty frequent follow-ups. I think they follow them almost every year. And so this is where the grant came in, because this data is super, super expensive. You can imagine that they put a lot of work into it. They don't give it out for free. So what you have to do to get this data is you have to send a proposal, and they decide if it's interesting enough or novel enough, and then you basically pick variable by variable what you want to study, and they charge you by the variable. So it ends up being like this costs like around $7,000. It's a pretty expensive data set to use. And within ALSPAC, this is where the epigenetics comes in. They were fortunate enough to have blood samples before people even thought of epigenetics as something to study. And so they had cord blood samples at birth, they had samples in mid-childhood, and they had samples in adolescence. And so they began to scan them. So they began to do the epigenome-wide profiling. And they didn't, it's expensive to do it, so they didn't do it on all 14,000 people. They picked a random subset of 1,000 children. And so they have 1,000 children where basically they scanned their epigenome and they have all the different levels of methylation at those CPG sites available. So this is great because that means I didn't have to do that. And there's a lot of processing that goes with data like this. And so basically this is what each EWAS looks like. So if we're talking about the one at birth, we have gestation from 0 to 40, and this is when they measured the variables. So they have 2 different time periods of life stress from the mom, and that could be intimate partner violence, that could be losing a job, that could be getting sick, that could be all sorts of things. And they measure it twice in the beginning of pregnancy and towards the end of pregnancy. And then they also measured mother use of alcohol, marijuana, and cigarettes. And this is really important, especially when we talk about prenatal stuff, because you can imagine drinking alcohol and smoking has a lot of effects on DNA methylation. And so we want to make sure that we're controlling, that when we look at it, we're not just looking at cigarettes really changing the epigenome. We want to make sure that we're isolating what we think is actually causing the different changes. And then same thing with adolescents. So they have a measure of different stressful experiences from age 12 to 16. And for adolescents, peer stress seems to be the most traumatic thing that you can look at. And they have really good measures of peer stress, so that's what part of what I'm going to be using. And they have depression, like I said, across these different time points. And they measure depression with the Short Moods and Feelings questionnaire. It's 18 items. It basically maps onto a depression diagnosis. And again, like for moms, they also have data on cigarettes and marijuana and BMI. And so those are the things that I wanted to control for adolescents, because research shows that adolescents who are depressed tend to smoke more, and adolescents who are depressed tend to have higher BMIs. Also, BMI changes. People with different levels of BMI have also different DNA methylation profiles, and then cigarettes and smoking affect methylation as well. So basically, you could have a third variable situation going on where you think it's depression, but it's really something else that's causing the changes. So I wanted a control for that as well.
Do you know whether they ask the question across like the 1,000 kids almost at the same time? Because if like ask me the question before the exam and ask him the question like after exam, probably like the answer will be totally different. But probably like he will have almost the same type of feeling like before the exam if you ask me that type of question.
I think the way that they ask is in the past 2 weeks. When we're talking about depression, they're asked like, in the past 2 weeks, have you felt this, and this?
Do you know like their, the date in terms of like when they ask the question?
Oh yeah, it's at the same time. They get a bunch of them to fill it out, largely in the same time points. Yeah.
You also have information about the genes per se, DNA.
Yeah.
And are you using that information? So I'm wondering.
Yeah,
Maybe
that's
like
a good
very
question.
naive question, but is it possible to distinguish the role between genes and the genetic and epigenetic effect?
Yeah. That's a really great question. That's a huge question in the research as well. Yeah, so not only do you have the SNPs, right, that vary between people, and so with a lot of this research, it's really hard to do that. And so what people usually do is they just take out the SNPs. So we don't look at CPG sites that are associated with any SNPs.
SNPs are.
Oh, sorry. The stands for single nucleotide polymorphism. So basically, that's just a fancy way of saying that there's a certain amount of genes that vary between individuals. So like I could have a CC and you could have a CT when we're talking about our genetic code, and that variation is called a SNP.
Okay.
Yeah, and so when it comes to methylation, there's a lot of studies that show that methylation is governed by the environment, but it's also governed by genes. So if you have a SNP, there's different levels of methylation. So like my levels of methylation with a CC might be different than your levels of methylation because you have a CT, and it might not have anything to do with the environment. So DNA methylation is under the control of both genetics and the environment, which makes it really difficult to study. And so the imperfect solution that lots of people have when they do EWASs is that they just remove all the CPG sites that are near or on SNP genes. Yeah. And so when we look, so this is the trajectory. So I basically wanted to see if I could find homogeneous subgroups in this population. So basically, these three groups, the individuals in them are very similar to each other, and they're very different compared to the other groups. That's basically what trajectory class modeling does. And these groups actually map on pretty well to what we see naturally occurring in populations. The percentage of participants that have high levels of depression kind of match what we see in the population. And these groups kind of match what other trajectory models have found when looking at depression. So a huge chunk of people have low levels of depression, around 77. And these people, which I find very interesting, the increasing group, they start in the beginning of adolescence, they start at really low levels of depression. So imagine the cutoff to be around here. Between 11 and 12. So they start really low, and they kind of skyrocket. By 18, they have really high levels of depression if like 11's the cutoff. So I'm interested to see what happened to these people. And then there's this group where I'm not really sure, I know what to call it. Yeah, I call it decreasing because when you map it, it looks like it's decreasing, but not by much. So you could either think about this as like maybe it keeps going down as time goes on, or it kind of is people who are always highly depressed before they started adolescence, and they still met criteria for depression during adolescence. So you can think of that group as kids who are just depressed starting and maintain that levels of depression. Yeah.
So the blue line, the increasing, that's 14% of the population.
14% of the sample. Of my sample,
sorry.
yeah.
Yeah, of your sample.
Mm-hmm.
And that blue line is measuring the average level of depression?
Oops.
What is that blue number? So at age, whatever, 12, where it says 5.
Oh yeah, so basically what this is, so when you do this late in class modeling, it basically puts everybody in a class. So you have like 1,000 participants, and it basically fits a model to see, can we pick meaningful groups out of all these participants? And so it classifies everyone. So it put everyone in one of 3 categories. So 14% of my sample is in this category.
Right. But now the blue line is measuring a depression index, a score.
Yeah.
And that score is, and it's rising.
Yeah.
And what is that score the average of the score across that 14% of the sample?
Yeah.
Ah, okay.
Yeah.
And so that.
But it's taking into account across time as well.
Yeah. So because it's rising across
Yeah.
time.
Yeah.
What are the underlying components of the mixture?
Oh, I'm not sure.
And the reason I bring that up is that you could imagine 2 groups of individuals. This is an example in which between 11 and 18, there's no growth. one of them's like this, and the other one's like a nice sine wave.
Yeah.
And so I think, just a thing to think about is these two types of exercises, they're parametric. In other words, they take a certain function.
Yeah.
And they ask, how would I sort the data into different parameters for the function? Whether these are dependent on function you work with. And what's masked here are higher moments.
Mm-hmm.
And at least, my view of adolescence, both participant and observer, is the average.
Mine too.
Yeah, the mean is not sufficient. In other words, you might want to think about alternative choices
Yeah,
for the trajectory models.
That's a good idea. I think I'll definitely look into that. And so the final samples look like this. And so I only took people that had both methylation and depression data. And so some people didn't have both. And this is the breakdown of what the trajectories look like. And then this is just some descriptive statistics. So most mothers were around 30 years old. They were full term. 100 of them smoked pretty heavily during pregnancy. And then lower amounts of numbers had moderate to severe marijuana use or alcohol use. And then the same thing with adolescents. Around 100 of them tend to be heavy smokers. And what's great about these two samples is that they have a high level of overlap. So basically, where we're looking at almost the same participants. So 801 of these participants overlap, which means what we can look at those 801 and see the methylation over time. And so going into the data now, so that's the data set that I'm working with. Let's go into a little bit about how you look at methylation and what goes into it, because it gets really, really complicated. And I was lucky because ALSPAC's data comes already processed, so it's great. But if you were to do this yourself, you would have to get a wet lab, and you would have to have people do a lot of this processing. And so basically, I would say almost all of the research that is done on epigenetics, especially EWASs, are through Illumina. So Illumina is a biotech startup that is the front runner of developing a lot of this DNA methylation technology. And if you're doing any kind of DNA methylation work, you're probably going to have to buy their technology. It's almost like they have a monopoly on a lot of these. And so they built these arrays that scan the epigenome, and they're manufacturing them really quickly. And so the problem with a lot of Illumina stuff is you have a study that uses an array, and then a year later, they're like, never mind, we have a new array. And it's really hard to match samples because they're scanned with different arrays. But they move really fast, so they keep improving their arrays. So there are 28 million CpG sites in the epigenome. There's a lot of them. Illumina does not scan all of them because it's expensive. They scan a specific subset. So they have scientists and researchers basically pick the CpG sites that they think might be the most important. And they focus on what I talked about, the islands, the shores, the shelves. So they don't really look at that random CpG site out in the code. They really focus on what they think might be important. So the Illumina that ALSPAC used looked at 485,000 sites. Now they have Epic, which looks at 850 sites. And there are technologies that look at all 28 million. You can do that, but it's like $1,000 per participant. It's very expensive to look at every single CpG site. But I think as technology advances, it's going to become less and less expensive to do that. And when you actually look at whole genome sequencing, the important genes, they're pretty much highly correlated. So this is a reliable way to get a good subset of the epigenome without having to scan all of it. And the way that they scan it is really interesting technology. So they use B-chip technology. And so the way that they do it is they basically create a SNP, and then they measure that SNP. So this is what a bead chip looks like. It looks big, but it's really small. It kind of fits in the palm of your hand. Each square represents a participant. So each square has a participant's DNA in it. And it usually comes 6 by 2. And so basically what they do first is they take the human DNA and they bisulfate convert it. And so when they do that, they create a SNP. So all the unmethylated cytosine bases turn into a T, and all the methylated cytosine bases stay the same. And so then you can actually measure the difference. So which bases converted and which bases didn't. And the way that they do that is on this bead chip, they have synthetic DNA. So they create DNA that is supposed to hybridize with the human DNA. And
Sorry?
yeah, go ahead.
I'm really confused.
Yeah, it's very confusing. I barely understand it myself.
With what you said previously, that you will get rid of the SNPs, you said before you are not, you want to look at sites that are far away from.
Yeah, you get rid of the SNPs in post-processing. So you get rid of them in the data. Yeah, you don't get rid of them in the processing part. Yeah, so once you get all the data, you can basically, since all of them are annotated, you can go and be like, okay, these are the SNP CPGs, let's just delete them from our data. Yeah. And this is what comes out. So basically they use this laser that scans it, and it shows up red or green fluorescence based on levels of methylation. And then they use a computer that quantifies the amount of changes, the amount of fluorescence, the ratio of green fluorescence to the sum of the green and red. It's very complicated. I'm pretty sure I barely understand it myself. Methylation is a zero-sum game. Basically, like either a CPG site is methylated or it's not. You don't get like a half methylation. But in a lot of studies, you'll see proportions or percentages of methylation, and that's because there's a lot of error in doing this type of study. So they have several copies of the same CPG site, and you just take the average of them. So before you can look at, so they scan this, then you turn all of these fluorescents into beta values. And then they do a bunch of quality checks. So they have to make sure that everything worked the way that it was supposed to. The first thing they want to make sure is that the participants are the same in the array. So because these are hand pipetted, right, human error can happen, and so people can put a participant in a different well than they were supposed to, and so they basically genotyped a handful of participants, I think like 80, and then they were making sure that their genotypes, their SNPs match up. And they also look at sex mismatch, so they looked to see chromosomal DNA to see if they're matching. So if on your phenotype data, it says that participant 192 is female, but you look at what's on the DNA data and it doesn't match up, then you have to get rid of that participant. You have to get rid of that sample. So they do a lot of quality checks, and they get rid of lots of samples that don't match. And there are some samples that don't match. There's a lot of human error that goes into this. And then they have control probes on the array that make sure that the technology worked. So all the different stages that goes into processing the DNA, they have control probes. And if those control probes showed that, for example, the staining of the fluorescence or the hybridization didn't work well, then we remove those probes. And then the important thing in doing methylation is batch effects. They're a huge problem for looking at this stuff. There's a lot of batch effects. So, a bead chip has 12 samples. Those 12 people look more similar than the next 12 people, so there's a lot of batch effects. And that could be for various reasons, right? The temperature of the lab, the time of the day, how long it was in the oven. This batch was in the oven longer than that one. So there's a lot of batch effects that we need to correct for them. So through all that, I didn't have to do all that, which is great. This is what you end up with. So this is what Alspec sends you. They send you just a text file. These are all the CPG sites. And then this is participant, and this goes down 500,000 down, right? So you have all the CPG sites, and these are the levels of methylation at each site. That's one file. And then the other file is basically how you identify. So each slide is one participant. This is where they were located on that bead chip, and that's the plate number, so the batch that they were part of. So this is what you get from Alspec. So this is what you start with, but then how do you analyze it? You need to have a really good understanding of R. And again, this is where the grant came in because I did not have a good understanding of R, let alone a good understanding of R to run a complicated EWAS. So I went to the same workshop twice. That's how complicated it was. I went 1 year and then I told my advisor, I need to go again because I don't remember anything. So I went to this workshop twice. It's a very, very good workshop at Columbia, also very expensive. So that's where the grant came in. And it basically helps you learn R and how to run these studies. This is actually my code. It's really complicated. I'm not going to go through code because that's boring. But you really have to, the only way to run an EOS is through R. It's like the only statistical program that has the packages, and that makes things easier. And it's free. All the packages are free. And this is what we talked about, removing additional probes. So we do remove the SNP probes because we have to make sure that the DNA methylation aren't due to genetics. We remove cross-reactive probes. That's just probes that tend to bind to multiple sites. And we have to account for race because different races have different DNA methylation profiles. Bristol is a pretty white place. There was only like, I want to say 25 non-white people in Bristol, so that was kind of easy for us. I just removed them from the analysis. And another important thing when we talk about epigenetics is cell type correction. So most EWAS studies uses samples from blood for analysis. But the issue is that every cell type and every blood cell types has their own epigenetic signature, like we talked about through cell differentiation. So we have to correct for the different cell type proportions. So ALSPAC, thankfully, has the proportions of all these blood cells in each individual, so you can correct for the proportions when you run your models. So this is a really complicated project. I don't think I realized how complicated it was. You need a lot of processing power to run this analysis. So if you think about it, there's, let's say, almost 500,000 CPG sites per person. There's around 1,000 participants. So you're doing that many linear equations. So it's a linear regression when you do an EOS times 6. Because I'm looking at 3 trajectories at 2 different sensitive time points. A single computer, even a couple computers can't run this. So basically you need a computing cluster. And so basically what a computing cluster is basically you have a central administrative node that basically takes your code and then sends it to all the different nodes and they run it simultaneously. So an additional thing that you need to learn is you need to learn how to code Linux because at University of Pennsylvania, their cluster is Linux-based. And so that's what the interface looks like. This is my code. And basically, you have to learn how to code Linux to run this cluster. And all you have to do is send your code to them, and they run these 500,000 regressions pretty quickly. So that's where the data is right now. So that's where I'm in. I don't have the analyses from this yet. So right now, the data is in the cluster, and it's running right now.
Sorry, you mentioned 500,000 linear regressions. Could you describe what each regression is doing?
I have no idea.
Oh.
So what happens in EWAS, it's a package. And so the code for running an EWAS is literally like a line. You just go EWAS, your independent variable, your betas, and then it runs it. So I don't really know like what each is doing.
The slide before, because you're doing it for each individual and for each CPG site. So I was just wondering what's going on there. Like,
No idea. Yeah, go ahead.
A follow-up question. I mean, is it like what people do in genetics? So they construct some kind of like a polygenic score, some kind of a factor, or what exactly these regressions do?
Yeah. You can do a factor or like you mean like a polygenic score or?
Yeah. Is there something an analog?
Oh yeah, I should talk about that. Yeah. Okay. Yeah, I should have. I should have talked about what the output looks like. Yeah. So when you do this, you basically get an output of hits. So it basically gives you a list of CPG sites that were differentially methylated between your two groups. I don't know if that helps. And because there's multiple testing issues, we use like a Bonferroni correction for it. So it basically any hits that survive a Bonferroni correction are significant hits. And so then you basically say, this CPG site is differentially methylated in people who aren't depressed versus people who are depressed, that one specific CPG site. And then since it's annotated, you can go back and see what gene that CPG site is next to. And sometimes you get a cluster, right? So you get a whole bunch of CPG sites that are around the same gene. And then you could do a gene network analysis to see what genes are in this network. But depends on how many hits you get. I don't know if that answers your questions, but I don't really know the background of what this package does. The way that it's explained is that it basically runs all the regression. It basically runs a regression based on the CPG. So it's basically going through every CPG site and seeing if this site is different from this site across two groups.
That makes me wonder why it's per person, but that's okay.
Yeah,
I was just
I probably can't have a.
okay.
What do you mean by per person?
So you time 1,000.
Yeah.
The regression.
You're doing it for each participant.
Yeah.
According
Yeah.
to the slide. So
Yeah.
I'm just wondering how many. It's not that big of a deal. I mean,
No,
if
it's
you're,
fine.
If you're confused, but I mean, yeah. I was just wondering if you're comparing across two groups
Mm-hmm.
and there's multiple people per group, then there's going to be multiple data points within each regression. So that was just my concern.
Okay.
But that's what I was confused about.
Okay. I wish I could answer it better. So then, yeah, you move it through the cluster. So basically, this is just the setup of what I've done so far on this project. So first, I found the longitudinal sample. I got the proposal approved. I looked through the variable catalog, which took months because they have thousands and thousands of variables. And so basically, I was reading the questionnaires over and over again to see which variables I think I could use. I requested the variables. I paid for them. I created all the covariates. I created the trajectories, the final sample. I learned how to write R code, which I didn't know how to do before. I learned how to write Linux code, and now the data is being run through the cluster. And this is probably this is the future analyses. So basically, I'm running an EWAS between low and increasing groups, low and decreasing it, and then the decreasing versus increasing, controlling for all these things: birth weight, maternal age, gestation length, substance use, and then plate ID for those batch effects. And then what you get from the EWAS is CPG hits that survive corrections for multiple testing. And then you could further go through gene network analyses to see. It depends on how many CPG gets. Some people get like 60, and then you annotate them, and then you see this clump of CPG is next to this gene, that clump is next to that gene, how are these genes related? And you could create a network analysis of what genes do you think are being implicated in depression.
Just based on proximity.
Yeah.
Yeah.
Yeah. So the CPG sites are located relatively close to the promoter areas of genes, so where transcription happens. So yeah, they basically annotate the proximity to the nearest gene. So I just want to thank you for the grant, for the training that it gave me, for understanding epigenetics, and actually it was really instrumental in getting my second grant, which is an NRSA, and I don't think I could have gotten that one without the R24, so I really appreciate that. Okay. Thank you. Does anyone have any questions, comments, suggestions? Okay. Very good.
Do you also have information about the mothers?
Yeah. So we have.
The epigenetics
we have epigenetic, but that's like beyond what I'm gonna do. But yes, they also have blood samples from mothers from when they were pregnant and at follow-up points. So there are studies that are looking at the co-methylation between mothers, especially in birth. So mother's methylation at birth and child's methylation at birth. They're looking at that and prenatal stressors or prenatal insults and seeing if those things are related altogether. And they also have fathers. So they also have father methylation data as well. And father SNPs and mom SNPs, and yeah, they have a lot of data. Okay, thank you.